This presentation will describe the Phenotypic IROA protocol, which is used for metabolic profiling experiments where it's not practical or possible to label the experimental sample. For example, when working with tissue samples, or measuring cells or spent media from large fermentation runs, or material from field-grown plants. In these cases, an IROA-labeled internal standard is generated, which most closely resembles the metabolome to be measured. Therefore, two IROA protocols are possible. The basic IROA protocol, which is an untargeted method, whereby control samples are labeled with 95% C13 media, and experimental samples are labeled with 5% media. And a second protocol, called the Phenotypic IROA protocol, a complex targeted method where control samples are labeled with 95% media and compared to their natural abundance counterparts. For both protocols, the IROA Cluster Finder software performs a full analysis, removes all artifactual peaks, and identifies and quantitates the remaining metabolite peaks. In the Phenotypic IROA experiment, the control or standard is isotopically labeled using IROA C13 media. An ideal standard is one that represents the entire metabolome of the fluid or tissue under study. For example, if interrogating the metabolome in pancreatic tissue, identification of metabolites can be achieved using an IROA-grown pancreatic cell line as a complex internal standard. The pancreatic cell line is grown in IROA mammalian media where all carbon sources are labeled with 95% C13. After 5 cell doublings and complete labeling of the cells, a complex internal standard is generated. The complex internal standard is then spiked into each experimental sample and processed as a composite, which removes technical variability. During the phenotypic experimental analysis, the Cluster Finder software will use the C13 IROA peaks in the labeled control sample to find and identify their associated natural abundant peaks in the experimental sample, allowing for a complex targeted analysis. Using the basic IROA protocol, where both the control and experimental samples are labeled with 95% and 5% C13 media respectively, the IROA signal as seen in this slide is made up of two halves, the C13 half and the C12 half, in which the carbon envelopes are mirror images of one another. When using the Phenotypic IROA protocol, the experimental sample is unlabeled and contains only natural abundance carbon. Here the Cluster Finder software identifies the 95% C13 labeled peaks from the internal standard and then calculates where to find the corresponding C12 peaks. The pairing makes the identity of all peaks measured unambiguous. Using an IROA labeled standard, all of the peaks may be identified according to the presence of their characteristic M-1 peaks. The natural abundant peaks corresponding to each standard peak may be readily identified because even though they do not carry an isotopic labeling, their exact mass and position are established relative to the standard. The distance between the paired base peaks defines the number of carbons in each molecule. If the pooled standard is well characterized and the compounds that are present in it have already been identified, then all of its compounds may be used as a point of comparison. Artifacts will have no match in the standard and are eliminated by Cluster Finder software in the final data set. In the phenotypic experiment, all experimental samples are measured relative to the standard. Therefore, the phenotype is defined by the deviation from the standard. For further information, please refer to the IROA website, iroatech.com, or write to info@aroatech.com. Thank you.